LSD cap. It should feel like a fairly comfy hat on the brain, right?
And so you can see everything that's going on in my brain in terms of emotional responses and...
I try and leverage what the sensory cortex can actually give us.
Right.
Because often what I'm looking at is the very molecular changes in the brain.
Just with our scientific collaborators here in the laboratory, looking at there's about 63 samples of LSD in these small vials in this piece of equipment here, and we're doing quantitative analysis. Quite a complicated and involved bit of equipment, but it's fascinating, the clinical research that's going on here to treat some of these mental health conditions. So this is where we record a microdose, is that right?
Yeah, weighing of LSD microdosing.
So it's very sensitive.
Yeah, it's a very sensitive product.
So we took samples at half an hour, 1 hour, 2 hours, just to see how much drug and how it was changing over time. But we also collected serum and plasma, which would allow us to look at things like proteins and cytokine changes. So we can look at plasticity factors, so that thing like BDNF. We can look at inflammatory cytokines. We then took another type of tube for analysis, so they've been partitioned out. So they are whole blood with concentrated to the white blood cells that would allow us to get a really nice DNA sample to look at genetic reasons why people might respond differently to LSD. The signal that's coming through your brain, I want that to be as loud as possible. And so what I would do is just put a small amount of gel under each electrode with the syringe here, push it in, and then just kind of scrape your scalp and, until the entire cap goes green.